Alright, so our last unit ended on page 41. That was the last page that you wrote on. So on page 42, we're going to leave this page blank. That's going to be our in-between units page. And then on page 43, this is going to be your cover page. Our next unit is on cells, which means that we're actually going to start writing today on page number 44. So please open your notebooks to page 44. And the topic for today is cells. All right, first things that you need to know about cells, there are three sort of rules that we're going to follow. The first rule, all living things are made of cells. So if it is a plant, an animal, a fungus, a bacteria, if it has the characteristics of living things that you learned in like 7th grade, so if it can reproduce, if it can take in energy and nutrients, if it can move, all of those things mean that it's living, and if it's living, it is made up of cells. Now, it could be made of a single cell, or it could be made of multiple cells, but either way, it has at least one cell if it's a living thing. 2 says that cells come from other cells. And we'll be learning more about this process later. This is the process of cell division. And it basically just says that cells can't come from nothing. Cells have to come from other cells. And that way they have all of the structures of cells, and they can copy their DNA, and they can make sure that the cell is fully functioning as a living thing. All right, finally, all cells are made of structures. That allow the cell to function. And you might be thinking of that song, cells, cells are made of organelles, try to pull a fast one, the cytoplasm gels. Now, some of these structures are actually organelles, but some of the structures are not organelles. So an organelle has to have a membrane around it. So a structure like a ribosome or a cell wall or a flagella, those are structures that do make up the cell and they do allow it to function, but they are not actually organelles. So sometimes these structures refers to organelles and sometimes it doesn't. All right, we're going to be going through a bunch of cells, and we are going to start with a bacteria cell. So we're going to start with this guy here. So make sure that you have this ready to go. But I'm going to start with just a note first. So this is my prokaryote. And this word refers to cells that are normally bacteria, but they are cells that don't have a nucleus. They don't have their DNA surrounded by a membrane. They actually don't have any organelles because they don't have anything surrounded by a membrane that's an internal structure. So we're going to define this. Well, first I'm going to put a box around it just to make it clear. That's our heading here. That these are things that have no membrane-bound organelles. Specifically, they don't have a nucleus. That tends to be the membrane-bound organelle that most people think of when they think of prokaryotes. This word actually refers to the fact that when we first were able to look at cells through a microscope, they basically looked like a soft-boiled egg. So it had the area around the outside that's the white, and then it has the yolk in the middle, and that yolk is the nucleus. And when they looked at prokaryotic cells, they said, you know what, there's no yolk. It doesn't look like a normal egg. And so this word actually, the pro part means before, and karyote actually means egg. So before they looked like an egg, they looked like a prokaryote. These are organisms that are unicellular. Now, uni means one, so like a unicycle has one wheel. So unicellular means one cell. So we're just going to define that here. Made of one cell. And another thing about these prokaryotes and bacteria in particular is that they have a type of DNA called chromosomal DNA. And that means that their DNA forms a chromosome. Again, we're going to learn about that later. But their chromosomal DNA is circular. It's really sort of in like one loop. And then they also have something called plasmid DNA. And plasmid DNA is really, really, really useful for us because that is how we can give other organisms new genes. So your DNA has your genes, and plasmid DNA is extra DNA that's inside of the cell, but it doesn't necessarily make up part of the chromosome, the things that are required for that organism to live. It's extra genes, which can give it extra functions. And we're going to do a lab later this year where we take plasmid DNA and we put it into bacteria, and the plasmid DNA has a gene that makes the organism have a fluorescent pigment, and so it's going to make our bacteria fluoresce in different colors, and we're going to be able to do that with plasmid DNA. Okay, now is when you need this bacteria, and I'm going to arrange it sort of in the middle of the space that I have left. So I'm going to tape this all the way down, taping the edges so that I have as much space to write as possible. So we're going to need to label the structures that make up this example of a prokaryote. This is a bacteria. All right, so first of all, we're going to label that chromosomal DNA. So in this example, this is my chromosomal DNA. And all cells have chromosomal DNA. So I'm going to put a star next to this to indicate that all cells have this. You have chromosomal DNA. The difference is you're not a prokaryote. Your chromosomal DNA is not circular. Oh, sorry, you guys can't really see that very well, can you? Okay. Another thing that this bacteria has is it has that plasmid DNA. So these circles here, they're different sizes. These are different plasmid DNA. So they have genes, they are useful, and human cells do not have plasmid DNA. The next thing we're going to talk about are the ribosomes. So ribosomes are super important. They're these dots here. So my ribosomes are important because they actually are responsible for making proteins. And if you remember from our previous unit, proteins are the doing things of the cell. So all of your cells have their characteristics because they have specific proteins. If we didn't have ribosomes, we would not be able to make those proteins. Our cells would have no function, they would die. All living things, all cells, have ribosomes. So let's give that one a star as well. And you're going to need to know that ribosomes make proteins. So make sure you're paying attention and copying these notes down. All right, the next thing on the list is the cell membrane. So this bacteria cell has an outer area, that's the cell wall, and then inside of the cell wall there's this darker gray cell membrane. So I'm doing my best to make it so that my arrow is pointing at the edge of that there. And I'm sorry, I cannot zoom in and record. A problem that I just realized. Now, my cell membrane has a specific job. All cells have a cell membrane, so let's give it that star. And the job is the same for all cells. And the cell membrane controls what goes into and out of the cell. It's kind of like a gatekeeper. So controls what goes in / out of the cell. So some things are able to go right through the cell membrane pretty easily, like oxygen, carbon dioxide. Those are small enough things that they can go right in and out of the cell. But other things like our monomers, like glucose, they can't fit through the cell membrane. So there are actually these proteins that sit on either side of the cell membrane and make sort of a passage. And then the cell membrane can open and close those to allow either more glucose to get in or more glucose to get out. So the cell membrane is going to control what goes into and out of the cell. All right, let's take care of this other outer layer. It is the cell wall. Not all things have a cell wall. So the cell wall, its job is to provide structure and support to the cell. Oof. Ooh, not my best notes there. And this outer layer is the cell wall. Now, we'll find out that in addition to our prokaryotes having cell walls, there are some eukaryotes, that's the other type of cells, particularly plants, that also have a cell wall. But the cell wall for plants and the cell wall for bacteria are made out of a different material, but it still does the same thing. So it still provides structure and support to the cell. There are just a couple more things that we need to talk about, one of which is this fluid that these structures are floating in. And so I'm drawing a box to indicate that this line is referring to that fluid. And this is the cytoplasm. Now, technically, cytoplasm refers to the fluid that organelles float in, and prokaryotes don't actually have organelles because there's nothing here that's bound by a membrane. So technically, they have something called cytosol. But we're just going to call it cytoplasm because it's the same stuff. It just doesn't have the same stuff floating around in it. So it's still the same thing. So we're going to call it cytoplasm. And cytoplasm is just a gel-like fluid. And that is also going to be found in all cells. So there's our cytoplasm. And last but not least, we have this tail thing. This tail thing is called a flagella. And the flagella is not found on all prokaryotes, but some prokaryotes do have it, and it provides locomotion. And I'm just going to say it provides for movement, because that's an easier word. So this is your flagella, well, the bacteria flagella. And again, not all bacteria have a flagella. There's one type of human cell that has a flagella. It is sperm. So the fact that sperm have a flagella allows it to swim in order to find an egg. It allows it to move around. And some bacteria have 100 flagella, sort of all over the cell. And those, they can move well. Sometimes it becomes a problem though. So more flagella, not always a good thing. All right, the last thing that we are going to put on this page is we're going to put a star to tell us what all of these things that we starred are. And so we're going to say here that all cells have these components. And when I say all cells, I'm talking all cells. So your animal cells, your plant cell, your bacteria cells, they are all going to have chromosomal DNA, some sort of a cell membrane, ribosomes, and cytoplasm. All right, I'm going to finish taping down the rest of this page, and then this one is done, and we will move on to our plant cells.